Hello, 25th August is the birth anniversary of Sir Hans Adolf Krebs. He was the German-born British biochemist. In the year 1953, with Fritz Lippmann, he received the Nobel Prize for Physiology or Medicine for discovery of the series of chemical reactions known as tricarboxylic acid cycle or citric acid cycle in the living organisms. He was the pioneer scientist in the study of biochemical pathway in the cells for production of energy. He is best known for his discoveries of citric acid cycle and urea cycle. In eukaryotic cells, the citric acid cycle takes place in the matrix of mitochondria. In the prokaryotic cell, these steps takes place in the cytoplasm. The citric acid cycle is a closed loop. The last part of the pathway reforms the molecule used in the first step. The cycle includes totally 8 major steps. The cycle can make a lot of ATP molecules indirectly by the NADH and FADH it generates. The NADH and FADH are electron carriers and they are going to connect with the last portion of the cellular respiration, depositing their electrons into the electron transport chain to drive synthesis of ATP molecules through oxidative phosphorylation. Interplay of molecule of acetyl-CoA is serving a lot to orchestrate the citric acid cycle through working as a feeding molecule. This acetyl-CoA can be obtained from pyruvate derived from glycolysis, from beta oxidation of fatty acids, from ketone bodies, and from amino acid metabolism. The oxaloacetate and acetyl-CoA are the two reactants to initiate the citric acid cycle. The enzyme required for the first step of cycle is citrate synthetase. In the first step of cycle, the citrate synthetase enzyme helps transfer the 2 carbon acetyl group from acetyl-CoA to the 4 carbon acceptor compound oxaloacetate to form a 6 carbon compound, the citrate. The type of this reaction is the aldol condensation reaction and it is irreversible. This reaction extends 4 carbon compound oxaloacetate to 6 carbon compound citrate. The second step of this cycle is the isomerization. Citrate is the reactant for the second step of the cycle. The enzyme involved in this cycle is aconitase. The step of citric acid cycle is with 2 reactions. In the first reaction, through the dehydration, citrate gets converted into cis-aconitate. The enzyme aconitase catalyzes the reaction. In the second reaction, through the rehydration, cis-aconitate gets converted into the isocitrate. The enzyme aconitase catalyzes the reaction. Thus, in the second step of cycle, citrate is isomerized by dehydration followed by rehydration to isocitrate. The enzyme aconitase catalyzes the two reactions using 6-aconitate as the intermediate compound. This reaction is a reversible isomerization. Isocitrate and NAD are the reactants for the third step of cycle. The enzyme isocitrate dehydrogenase is required for the catalysis. Here, NAD stands for nicotinamide adenine dinucleotide. This third step of citric acid cycle is also with 2 reactions. The first reaction is dehydrogenation, in which isocitrate gets converted into the oxalosuccinate. The second reaction is decarboxylation, in which the oxalosuccinate gets converted into the alpha ketoglutarate. This third step of cycle is oxidation type. It results into formation of 1 carbon dioxide, 1 hydrogen ion and alpha ketoglutarate. Here, isocitrate dehydrogenase is allosteric enzyme. Therefore, the third step of citric acid cycle is a rate limiting. The reactants for the fourth step of cycle are alpha ketoglutarate, NAD and CoA. The enzyme involved in this step is alpha ketoglutarate dehydrogenase. The fourth step of a cycle involves decarboxylation reaction that transform alpha ketoglutarate, NAD and CO into succinyl-CoA, NADH, hydrogen ion and carbon dioxide. This reaction is irreversible and with exclusion of CoA. The reactants for the fifth step of cycle are succinyl-CoA and GDP. Here, GDP stands for guanosine diphosphate. The enzyme involved in this step is succinyl-CoA synthase. The fifth step of cycle involves a substrate level phosphorylation reaction that uses succinyl-CoA, GDP and P1. The step is yielding succinate, CoA and GTP. The GTP is later used for ATP generation. The reactants for the sixth step of cycle are succinate and FAD. Here, FAD stands for flavyl adenine dinucleotide. The enzyme involved in this step is succinate dehydrogenase. In the sixth step of cycle, the succinate is converted into fumarate. Here, the FAD is used to convert into FADH2. Next one. The reactants for the seventh step of cycle are fumarate and H2O. The chemical reaction is of hydration type. During the seventh step of cycle, hydration reaction combines fumarate with water to form malate. The reactants for the eighth step of cycle are malate and NAD. The malate dehydrogenase is enzyme responsible for catalysis. During the eighth step of cycle, oxidation reaction converts malate into oxaloacetate. The oxaloacetate is used again to continue the citric acid cycle. Here, third molecule of NADH is also produced. The NADH accepts the electrons which allow production of ATP. The NADH, GTP, FADH2 are the electron acceptors and allowing ATP production. Thus, in citric acid cycle, 24 ATP molecules are produced from single molecule of the glucose. Through the process of glycolysis, citric acid cycle, electron transport chain, 30 to 38 ATP molecules are produced from single glucose molecule. Likewise, amino acids enter into the citric acid cycle. Fatty acids can also be broken down into acetyl-CoA that can be used for starting the citric acid cycle. Proper name for citric acid cycle is the cyclic oxidation of the substrates. However, many people refer to the process as Kreb cycle in the recognition of contribution of Hans Kreb to the discovery. Kreb discovered the formation of citrate from oxaloacetate and pyruvate, the missing link that allowed the known reaction to form the cyclic sequence. Pyruvate clearly entered into the cyclic system of oxidation of substrates. It wasn't established until later that citric acid was indeed the first substrate formed from the reaction of pyruvate and oxaloacetate. So the cycle was called simply the tricarboxylic acid cycle for many years. Both the names are accepted as along with the term Kreb cycle. After the string of recent successes, journey of global biological sciences guided by Sir Hans Adolf Kreb achieved in a qualitative applicable metabolism. His contribution tried its best for giving a significant dimensions to the global biological sciences. Let us pray for this salvation of the departed soul of this great scientist. Thank you for participation in the celebration of day of birth anniversary of Sir Hans Adolf Kreb. Thank you.